thanks for the introduction. I'm founder and CEO of Interherence, and we basically develop tools that should help to improve workflows when dealing with high sensitivity microscopy. By that we understand any type of microscopy you do that is either sensitive to a single molecule level or deals with other very sensitive mechanisms or systems, as for example cells. Interherence itself is a startup, as mentioned. We are not that old. We are located in Erlangen. That's the center, basically the south center of Germany. We are funded by, well, several agencies. One is the Federal Ministry of Education in Germany, as well as the local Bavarian Ministry for Economics. I just want to mention this because I have to. We have roughly 100 customers worldwide to which we talk and who are already using our technologies. We are quite a diverse team, an international team of approximately 18 people currently. We are growing and we are also looking for people who would like to join us. So if you know someone who's finishing up his PhD or who's looking for a next step in his career, please let them know or forward them our addresses that we are actually recruiting specifically with background in microscopy. What we do and what we try to solve are the current pain points in microscopy that we foresee, and we group them into those 4 points. One is temperature control. It's not so trivial as one actually might think. The other issue is that the most of the setups that they're using in order to go down to the single molecule level can be quite complex and very sensitive to user inputs, and that's something that is not always wanted. And then on the other hand, you always generate a lot of imaging data that needs to be basically analyzed and features needs to be extracted. So with one of our products, that's the VAHIT system, we try to solve the issues that there are with temperature control in sample volumes that we foresee. And one might think that that's already solved, right? So you have this environmental box and you set the temperature to a certain value. But if you dig into the details and you actually investigate temperature sensitive processes and you try to track the temperature inside your sample volume, you will see that this is not that straightforward. You have many effects that can affect the sample temperature that you might simply not track with your current systems. And that can be based due to evaporative cooling of the liquid in which your sample is hosted. There can be a thermal drift in your setup that basically worsens your optical resolution. You have certain limitations in terms of temperature, either on the higher side or on the lower side of temperature, in which you can perform like super resolution studies. The objective itself is a very undefined element which can act as a heat sink. And overall, if you work at high temperatures and you don't do that well, you can have a strong degradation in your imaging quality, which you might want to avoid, specifically if you go to high resolution studies. So what we did is we basically, in general, the scheme of our company is that we exploit highly sensitive sensor systems that are incorporated on a cover slip. And that can be either electronics, as in this case with the VAHIT system, or it can be integrated optics, as in the case of the other system that I will also introduce you shortly to. So with the VAHIT system is our solution to temperature control in very tiny sample volumes that allows you, independently of your experimental setup, to track the temperature in your sample volume, also to control it dynamically. And it's kind of an add-on solution that makes this quite plug and play. The core of this product, of the system, is are these functionalized cover slips that I was mentioning. They are incorporating a heating element as well as a small temperature probe that is in direct thermal contact with your sample volume, and thus gives you feedback on the temperature independently of what you're doing with the sample. So if you have, for example, a perfusion system, or if the temperature simply changes in your lab, you can track the temperature of your sample volume and adjust for it that it stays at a given temperature under which you want to perform your experiments, for example. These cover slips are looking like that, so they're transparent, so you can use them with inverted microscopes, with oil immersion systems, but also with upright microscopes. They're quite versatile, and you can even put your own reservoirs on top or microfluidic solutions on top, depending on what you actually want to achieve or do in your experiment. The substrates are then simply put into a microscope adapter that is connected to a control unit via a connector head, and that then allows you already to read out the temperature and control it independently of what you're doing. There are multiple ways of how the system can be operated. I just want to show you a short video of that was recorded in a lab where we actually hooked up the system, we put it on a microscope, and we programmed a certain temperature profile through which the sample volume is cycled when this movie plays. And this is what you see here. So this green line is the target temperature of your sample volume, and the blue line is the actual measured temperature. I hope the video is working. Sometimes Zoom has some issues with lagging videos. But you see that the sample temperature basically follows instantaneously your set point, and this allows you to do some kind of thermocycling experiments, for example, on your microscope, without affecting the imaging quality at all. The system can be used up to 100 degrees C. At 100 degrees C, you have slight degradation of the imaging quality, but this is mostly due to your intrinsic properties, optical properties of your immersion medium that you're using, that might just induce some spherical aberrations in your systems. But the beauty is, one might think, where's that interesting to go to those temperatures, to those high temperatures, but there are several organisms actually that like thermophilic bacteria that like high temperatures and very strong or very aggressive conditions. And for these systems, for these biological systems, that's quite interesting. But also for DNA science or proteins, different temperatures play an essential role for their functionality. And if you want to quantitatively study those, it's quite important to stabilize their temperature, or to at least control the temperature or know the temperature. We also offer a system solution where you can go below room temperature. So the heat itself only allows you to stabilize the temperature of a sample volume above the surrounding temperature up to 100 or 200 degrees C, depending on the version. But we also have a new system that will extend that temperature range to also work below room temperature. For example, if you want to slow down certain biological processes, or slow down only your molecular diffusion or binding, unbinding rates, it might be crucial to decrease the temperature to, for example, 12 degrees C or 8 degrees C. And in combination with oil immersion objectives, that is also not that trivial. And for that, we have developed that solution that allows you to stabilize the temperature basically between 8 degrees C and 100 degrees C in combination with Vahid. Without Vahid, it allows you to stabilize the temperature between 8 and 45 degrees C. The beauty is that you can additionally control, well, your air that is basically going inside. This means the CO2 level or humidity, and then it's very nicely suitable also for lifestyle imaging experiments. The precision with which you can control the temperature is approximately 0.1 degrees C, which is quite good. That's the absolute values. The relative precision is much higher if you basically have a calibrated environment. But besides that, so this is just a short look onto the Vahid system. I want to tell you a bit about the new product that we are launching. And this product deals with simplifying the optics that are needed to do turf microscopy. So specifically using high-NA objectives and the alignment turf modules in the alignment optics that you require in order to hit the turf angle. To introduce this, I usually start with educating or reminding people of the history of turf, because the first turf implementation was not objective-based turf. Objective-based turf was only implemented, or the first commercially available objectives were ready in the late 90s. But the first implementation of turf microscopy, itself was prism-based turf. Back then Axelrod introduced this for biological applications, and this was a very neat method to excite sample volumes. However, you have certain restrictions when you do prism-based turf. Specifically, your sample access is highly restricted. But besides those two implementations that are widely known, there's a third way of doing total internal reflection microscopy, and this is waveguide-based turf. Also, this is an approach that has been already established in science for quite some time. The first papers that investigate this method for biological matters comes from the Plasmon community and date back to the 80s. In the 90s, there were also some commercial developments going on in that realm. But basically, starting 2015, more and more scientific groups started to implement their own waveguide-based turf systems for the specific applications. And this reasons is simply that the thin film technologies and the microfabrication technologies evolved in a way that it became accessible to more people to actually build these systems for their own applications for themselves. And we, Interherence, we are now the first company who's commercializing a waveguide-based add-on solution for basically any microscope, upright or inverted microscope, to equip it with turf capability without the need of modifying or changing, adopting any optics in your setup. And let me also remind you why turf is a great technique, or why is it an important technique to study systems. So turf has the unique properties that you can generate a very shallow excitation volume, small excitation volume, just very close to the cover slip. And this means that the rest of the sample volume, in which you might have cells or other organisms or systems, is not exposed to the light during the investigation. Thus, the method itself has a very low phototoxicity, as you do not illuminate, as in a confocal system or a wide field system, everything of the sample volume, even the layers which you're not imaging. Simultaneously, because you just illuminate a very shallow region, you generate a very high or very low background, because very little molecules can contribute to your signal, and those are the molecules of interest to you. That's why you end up with a very high signal-to-noise ratio. And this together is ideal for single molecule studies and super resolution microscopy. However, what you require with objective-based approaches is shortly sketched here. You need to come in with your beam in a certain angle that you hit the total internal reflection condition. And this arrangement can be nasty, as it comes with a lot of drawbacks, which I tried to summarize on the right side here. You are always required to use immersion medium in order to be able to hit the critical angle. You end up with a small field of views, maybe 60 by 60, 100 by 100 micrometers. The alignment can be tedious, it's high maintenance. If you want to study temperature-dependent things, as are often of interest, this gets very, very difficult, because as soon as you change the temperature, you can easily use your right excitation angle. The penetration depth is pretty undefined. You would actually calibrate it for each setup every now and then in order to quantitatively extract information. And often you suffer from inhomogeneous field distributions as well as poor polarization purities. And we think that those drawbacks, although you have huge benefits with turf, limit the field of applications and make it just difficult to perform quantitative and reproducible studies using turf microscopy. There are several papers in literature that are well aware of that and that try to, that educate people how you can improve your turf illumination, or what is important when setting up a turf microscope. A few of them I put here on this slide, but all these methods are working, but they require time. And not only time, they also require experience with the setup, which not everyone might have, or not everyone might have that time to actually perform these measurements or these studies before doing turf microscopy. And that's why we thought it's a great idea to start developing the product QSight that should make turf microscopy much more reproducible, as well as increase the usability quite a bit and extend it to people who are maybe non-experts with microscopy setups itself. So what is fundamentally different in our approach is that instead of using a light beam that is hitting the cover slip in the critical angle via an objective, we integrate an optical layer, a thin film layer on a cover slip that has a higher refractive index than the surrounding and thus supports an optical mode. And what we do then is we launch light into that optical mode that propagates along the waveguide. And we have chosen the waveguide mode in a way that it has a strong evanescent field tail that can enter into the sample volume that might sit above. So this should indicate some cells, for example, and if the cells are sitting then on the waveguide, they can experience the evanescent field, absorb the light, scatter the light, and re-emit it. And this is then your signal that you can collect with any objective, which sits below the cover slip or this waveguide chip. And one of the big benefits of this is that you in principle decouple the excitation mechanism. As you have here, you have the same optics for the excitation and the detection. And in the waveguide approach, you decouple that. You have this waveguide layer that generates the excitation, and you can now use independently of that any collection optics in order to capture your light. So the main benefits are exactly this. You fundamentally decouple the excitation from detection and introduce simultaneously a semi-monolithic structure for the excitation optics. This means that the way how you excite the sample is engraved in the class. It's defined by the micro and nanostructures on the chip that cannot be changed by the user. And thus, this means that it's everywhere the same, independently when you measure and independently where you measure. And this brings quite a bit of reproducibility. So this QSAT system should not only improve the benefits of TURF microscopy, so you can generate with this even higher signal-to-noise ratios because you don't have any backscattering from the collection optics. You have even lower phototoxicity because the excitation can be more efficient. But simultaneously, you work also on the shortcomings of TURF microscopy. Because now you can use any objective, you generate very large field of view in the range of square millimeters, which enhance your output tremendously. The system is low maintenance, it's self-aligning, it's temperature insensitive, so you can easily study temperature-dependent processes. You have well-defined penetration depth, which is only depending on the sample refractive index. You can generate very flat illumination profiles, which are determined by the waveguide properties, basically, and you end up with a very, very high polarization purity. And that is summarized in what we would name then a space-proof system, because it can be literally plugged onto any microscope without any change. And you just hit a button and then you have a very clean TURF illumination. So what does this large field of view mean? So this is a fluorescent image, example data of a cell, of a HeLa cell, which has a typical diameter of, let's say, 20 micron, while the typical field of view in a TURF microscope is 60 by 60 or 80 by 80 micron. But if you crank this up now to a few hundreds of microns by a few hundreds of microns, you can actually acquire these kind of data. And this was acquired with one of our chips or with one of our systems on a kind of not spectacular microscope using an air objective. So you see all these cells spread out on the chip being homogeneously illuminated via our waveguide. And that means you can just generate way more throughput, either in live cell imaging or in single molecule studies when it boils down to the number of molecules that you can count per time unit. The system itself is composed out of four main components. You have the core of the component of this system, as with the Vahid, are the Q chips. These are the cover slips, which have an integrated optical waveguide onto which you put your sample. So this can be your cell culture or your protein solution that you put on there or you prepare on these cover slips. Those cover slips then are inserted into a microscope adapter. This is a system that sits just on your translation stage. Your objective can then come from below or from top. A light coupling unit sits on this microscope adapter and ensures the automated coupling of the light that it hosts into the waveguide. And this light coupling unit is connected to a control unit, which is of the size of half a shoebox, let's say. And with that, you can just do turf microscopy wherever you want. That's how it looks like when it's mounted on a 2D translation stage. But most importantly, what kind of applications do we foresee? So we think that it can have a major impact on multiple in multiple fields. We come up from the single molecule community. That's where we basically have developed the system for what we have to develop the system for, and we see that it's a very nice system for these applications because you just generate high throughput with a very high signal to noise ratio without compromising on any resolution. So you can easily do super resolution storm type of measurements. But also for lifestyle imaging, often the data that you require when you do turf stem from multiple field of views, and that can be kind of time consuming to study multiple cells. And the system can just help you by increasing the field of view and studying either very large cells that extend over multiple field of views typically, or by just studying an increased number of cells. But also in general for biochemical assays, you can use the system for multiplexed analysis if you want to study, for example, concentration gradients, temperature-dependent processes, and so on and so forth. We will have this QSight system available for testing with a few reference sites via the Euro-BioImaging group, basically. If you're interested in getting your hands on one of those systems, either by HeatAerobox or QSight on one of the reference sites from us, please just feel free to contact me or one of my colleagues, and we would be happy to get you started to get your own experience on one of the systems. Thank you.